In this video, we will discuss about the effect of or mechanism of this AOH1996 that cause reduced metastasis in the cancer cells. So in order to do so, they decided to check the replication on the replication fork stalling, whether it has happened in the cells that are treated with AOH1996 or not. In order to do so, they use these two reagents, that is CIDU and IDU, which can be detected by different fluorescence. And before this AOH1996 introductions, they use CIDU so that the replications is shown in the green color, and after this AOH1996 or control DMS of treatment, they use this IDU, which is shown by which can be seen using different fluorescence. And then they check this ratio, that is CIDU by IDU, and if there is a replication fork stalling is happened, then less amount of IDU will be formed and we will see a high CDU by IDU ratio. And this is what we see in the dose dependent increase. The bottom side of the panel A shows the immunofluorescence image, where the red colors red fluorescence indicates that it is formed or it is replicate after this AOH introductions, and green fluorescence indicates that it is introduced before AOH1996 or control introductions. So this one is for control, and another one is the dose dependent increase in the AOH1996. We can see that with the increase in the dose, the green fluorescence is high and red fluorescence is lower. And when they quantify the ratio of the CIDU by IDU, they see that with dose dependent increase in the AOH1996, there is a decrease in the ratio of the CIDU by IDU, indicating that this IDU amount might be lower. So which is the indication that there is a replication fork stalling happens with dose dependent increase in the AOH1996. When they check this total number of these new origins, this percentage of replications events, and from this immunofluorescence image, they are able to find out that this replication events is increased with increase in the dose dependent AOH1996, but that increase is not statistically significant. So they call it as the mild dependent on this AOH1996 concentrations. Then they check the DNA damage using gamma H2AX, and they are able to see that with increase in the time period, there is an increase in the signals for the gamma H2AX, and the more predominant increase or the more significant increase in this gamma H2AX signal is for this MIA PACA2, which is a 68-whole-year pancreatic cancer cell lines, or pancreatic adenoma carcinoma cell lines, pancreatic adenocarcinoma cell lines from the 68-year-old patients. So they decided to go and do some detailed investigations on this MIA PACA2 cell lines, and so they provided the immunofluorescence image, where we can see that with the compared to the untreated with the 1 micromolar AOH1996, we see a more amount of gamma H2AX, more signal for this gamma H2AX, indicating the more DNA damage. And this is the quantifications which shows that with dose dependent increase in the gamma H2AX signals. And now they wanted to check the cell cycle assay, so that is where whether it has any impact on the cell cycles or not. And compared to the no treatment, with the treatment of this AOH1996, they are able to see that there is a cell cycle arrest at this G2 and M phase that can be seen here. With increase in the time, there is increase in the G2 and M phase cells that are in G2 and M phase. These things are quantified in the panel G, where we can see that with increase in the time, the G2 and M phase proportions of the cells increases, indicating that there is a cell cycle arrest occurs in this G2 and M phase. Now they wanted to check the apoptotic cells, or the cells that are under apoptosis. So for this they used the dose dependent study of this AOH1996, and compared to the untreated, they can see the increase in the apoptotic cells from 16% to 15% when they increase the dose of this AOH1996 from 250 nanomolar to 1000 nanomolars. And these things they have quantified in the panel I also, where we can see that the dose dependent increase in the apoptotic cells. So all this together, all these results together indicates about the mechanisms somewhat that AOH induces cell cycle arrest and also cause this DNA damage by replication fork stalling, and that triggers this apoptosis. That might trigger the apoptosis in these pancreatic cancer cell lines, and more specifically in this MIA PACA2 cells, which is a cell lines derived from a patient who is affected with pancreatic adenocarcinoma. So I hope you understood the somewhat these mechanisms about this AOH1996 on the pancreatic cancer cells. So if you need to understand more about this experimental results in detail and want to refer to some of the supplemental figures, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.